Welcome to this biochemistry lecture on glycolysis. So, as we discussed in the intro to catabolism that we had earlier, the lecture we had earlier, the video we had earlier, we discussed the general concepts of the whole pathways of catabolism. Today, right now, we are going to discuss glycolysis. And glycolysis sits right in the center of it. It's an extremely important part of the process, and we'll discuss the glycolysis and some of the pyruvate options that exist in there. As we noted earlier, we're taking lipids, carbohydrates, proteins, and we're going to feed them into glycolysis. And in the subsequent lectures, we'll talk about how each one of those biomolecules gets converted so that it can go through glycolysis and then go into the citric acid cycle and oxidative phosphorylation. Hopefully, this gives you an idea of what we're talking, where we're talking about in this grand scheme of things. So glycolysis, basically, the idea is we're going to take glucose as an energy investment, and we're going to convert it into 2 ATP, 2 pyruvate, 2 water, 2 NADH, plus 2 protons. And I may have said that where you may not have. So the net is 2 ATPs, 2 pyruvates, water, 2 NADHs, and 2 protons. That's the net. And we are going to use some ATPs along the process, especially in the first step. But so we have some energy investment in the first steps, and then we start paying off. We're going to convert it down into pyruvate. So we go from a 6 carbon into 2 3 carbon, and along the way we're producing NADHs, ATPs, and we're generally going to then feed into other cycles that can generate more energy out of these molecules than what this can, but we're still generating a net sum. Okay, so that's the general idea. Let's go in through the steps. The first step is we're going to take glucose, we're going to add ATP, so we're going to have to add energy in. We're going to have a cofactor of magnesium, and we're going to generate a glucose 6-phosphate and an ATP. So we're going to take the high energy ATP bond. Hexokinase requires it, and this is a surface representation of glucose 6-phosphate, and you can kind of see how nicely that glucose fits in there and what comes out. And there's a cofactor in there, and you can see all the residues that interface in there. I would encourage you to look this up on the PDB. It's the PDB model numbers right there, and you can actually go in and see exactly where that glucose fits in there. It's very, very encouraging step or enlightening step when you actually do that. And you can see what we're adding is the net sum of this is we're getting this nice phosphate group added to this sugar. That's step one. Again, it requires an ATP bond. One of the most common drugs that you're going to find out there is metformin. And metformin is a drug that inhibits ATP synthesis. So you can see one of its functions is it does that. So it fits right. It's a nice inhibitor of this hexokinase, and it fits right in there with hexokinase 2 in particular. And you can see in the C conformation. Sorry, in C, you can see where it fits nicely in there. Group A in this molecule is where the glucose sits. B is where the closed conformation of the molecule. So hexokinase breathes, right? And I wish I had an animation that I probably will try to generate one to show you later. But the A is in the open conformation, B is in the closed conformation, with glucose and glucose 6-phosphate highlighted in orange, or green and orange. And close C, section C is where metformin fits in, and you can see it's an induced fit. So it does change conformation, and then D is the close conformation of hexokinase in complex with glucose. and ATP, and magnesium is highlighted as well. So anyway, and then you have this you have the chemical structure of metformin. Metformin is an extremely common drug, and is used for all kinds of metabolic um, indications. All right, so step 2 of this process of glycolysis is we're going to take the alpha 1 or the glucose 6-phosphate that we just generated in step 1, and we're going to create a fructose 6-phosphate. And you can see this pretty readily in this boat conformation. The enzyme is a phosphohexose isomerase, and this is an isomerization reaction. As you can see, we're basically going to be moving the carbon, one of the carbon, into actually the number 1 carbon into a tautomerization. So the glucose 6-phosphate goes to a fructose 6-phosphate, and you can see that we also are going to go through an enediol intermediate in this process. And basically, the glucose 6-phosphate, we're going to add a keto intermediate. That's what step 2 is. It's a again, I would memorize the products, reactants, and the enzyme. Glucose or glycolysis step 3. We take what we just did, the fructose 6-phosphate. We're going to add another energy source in here, ATP. So this is the second step. You need to know that in step 3 we're going to be using an ATP. In step 1 we use an ATP. In step 2 we do an ATP. This is where phosphofructokinase comes in, and basically we're going to add another ATP. Anytime you add an ATP, it's a kinase. So we're going to take magnesium as a cofactor in there, and we're going to add another phosphate group on the number 1 carbon, and we're going to have ATP. That's step number 3. All right, step 4. We're going to take product of step 3, fructose 1,6-bisphosphate. This is the linear form of this. Normally we're going to be dealing with the ring or cyclic form of this. Anyway, we're going to take this and put it into an aldolase. The aldolase is going to break it into 2. So we're going to produce dihydroacetone phosphate and diglyceraldehyde 3-phosphate. So these might start looking familiar to you from other places that we've talked about in biochemistry, but basically what we're going to do is we're going to cleave this through an aldolase. So in 2 triose phosphates. Step 5, we take the dihydroxyacetone phosphate and we're going to convert it to the glyceraldehyde 3-phosphate. So we're going to isomerizing this triose phosphate. It's catalyzed by the phosphotriose isomerase. You're going to want to know that. And it involves 2 successive keto-enol tautomerizations. And so only the D enantiomer of glyceraldehyde 3-phosphate is formed. And so that way we're going to take, and everything is consistent. So step 5 is basically taking the hetero results and making them homo. So now what we have D-glyceride 3-phosphate as our products that allows us to go into the next step. D-glyceraldehyde 3-phosphate. We're now going to take NAD+, which is an electron carrier. We're going to go and we're going to add on and a phosphate group, and we're going to add the phosphate group onto the glyceraldehyde 3-phosphate, generate a 1,3-bisphosphoglycerate and NADH. And so the reaction is that we're adding on a high energy phosphate bond right there, and we're adding on electrons and a proton onto NADH. That's an important component of this whole process. And so the product is a phosphate ester and a high energy carboxylic phosphoric anhydride. So we've got these great products that are coming through high energy. All right, step 7. We're going to transfer the phosphate group from the 1,3-bisphosphate to ADP. This is an energy generation step. Sometimes you'll see it as GDP. Uh, um, depends on it's very specific to the type of uh, enzyme that's catalyzing the reaction and the location. You can abstract out that step 7 is we're going to take a nucleotide and we're going to add back in this phosphate from the 1,3-bisphosphate glycerate. Through the enzyme, the catalyzing enzyme here is the phosphoglycerate kinase. So we have an ATP, we're adding it, we're transferring that ATP from the 1,3-bis, one of the phosphates from the 1,3-bisphosphate phosphoglycerate to the ATP molecule, and we end up with 3-phosphoglycerate. That's step number 7. Step number 8, we're going to use a phosphoglycerate mutase to take, and we're going to have, with our 3-phosphoglycerate, okay, we're going to transfer that phosphate group to the C2 carbon of the triose molecule. And this is just a mutase. It's just going to ping it up. It's just going to move that from the C1 to the C2 carbon. Step 9 is a dehydration of the 2-phosphoglycerate, where we're going to take the 2-phosphoglycerate and we're going to, through an enolase and a magnesium cofactor, you're going to want to know both of those, we're going to make a phosphoenol pyruvate and water. So we are going to take off that hydroxyl group. The product is going to be water. Step 10, we then take and transfer that high energy phosphate to ATP, or ADP to make ATP. So we're going to take phosphoenol pyruvate through a pyruvate kinase, right? And magnesium, you might notice a really important trend here. Magnesium is an important cofactor in a lot of these reactions. Just as a side note, magnesium is also very important in things like DNA replication. Magnesium as an important cofactor is a very simple fix if some of you have like migraines. It's a very simple attempt to try, because a lot of people on our diets, they don't have enough magnesium. And we don't have enough magnesium through eating unprocessed food. Processed food has a tendency to take out magnesium. It's a very simple attempt to decrease things like migraines. Anyway, we take phosphoenol pyruvate, add another reactant is ADP. Now we're going to end up with pyruvate and ATP. Okay, so you might have remembered from the beginning step, pyruvate is an intermediate step that's going to lead to acetyl-CoA and then take us in through the Krebs cycle. And so that's where we are right now. We're now ready to go into an intermediate step right before we get into the citric acid cycle. So the net sum of these 10 reactions for glycolysis is we take glucose, we add 2 ADPs, we add 2 phosphate groups. Through glycolysis, we end up with pyruvate, 2 NADHs, 2 ATPs, 2 waters, 2 hydrogens. Actually, and 2 pyruvate. Sorry, I missed that 2 there. So that's the sum. So basically, we're taking a glucose, a 6-member carbon. We're going to end up with 2 3-member carbon. We end up with molecules that we end up with 2 energy molecules. Okay, bonus. That's energy generation. But we also end up with 2 molecules that are electron transporters and 2 protons that we're going to send through the other cycles in order to generate more energy for the cell. Again, here's kind of the steps, the energy yield. In the first one, in step 1, 2, and 3, in particular in step 1 and 3, we're going to use an ATP. So where net sum is 2. Phosphorylation at the end. This is at the end, right? So out of 1 glucose molecule, this is kind of giving you the idea, but we do have to go through other cycles. In 1, 2, and 3, we end up using 2 ATPs. Phosphorylation, we produce 4. That's after we go through all the cycles. 6 and 9, we end up with a phosphate transfer to ATP. There's another 4. Oxidative decarboxylation, that's where we take the 2 pyruvate and generate acetyl-CoA out of it. That's later in the process. That's 6. And when we do oxidation to 2 acetyl-CoA in the citric acid cycle, we end up with 24. So the total out of one glucose molecule is 36 ATP. Now the net out of just glycolysis is 2, but we also end up with pyruvate and a few other cofactors. You can see now that when we take glucose, we can generate a ton of energy. Again, look at this thing from the big picture. We see where we have glycolysis that sits right in the center. And the important parts of this thing are that it's going to feed through pyruvate and acetyl-CoA into the citric acid cycle. And that's where we're going to have the convergence. Now, tomorrow or in the next lectures, we'll talk about lipids, carbohydrates, and proteins and how they feed into the glycolysis and citric acid cycle. But for now, it's important for you to know where this kind of sits in the grand scheme of things. Now, we're also going to talk about catabolism of glycerol right now. Glycerol, as you might note, very briefly, glycerol is an important molecule in fatty acid catabolism and beta oxidation. And so we're going to take, and this is just the glycerol enters the glycolysis via the dihydroxyacetone phosphate step. And we basically take glycerol, we use energy to convert it to glycerol 1-phosphate, and then we take, and then another step, we create dihydroxyacetone out of it. You can see how it's very similar to glycerol in this process. And from there, it can feed into the pathway. And so it can feed into glycolysis at that important step. Okay, we also have ketone bodies. Oh, sorry. We also have pyruvate that we need to talk about pyruvate. Pyruvate, we'll talk about some of these other steps in a little bit. Pyruvate is the end product of glycolysis, but it's also an important step in other energy generation. So obviously we could take this, go to acetyl-CoA, go to citric acid cycle. That's great. In anaerobic conditions where we have when you're like exercising and contracting muscles, we can also generate lactate out of this. And the other thing that can happen is in anaerobic conditions in yeast, we can generate ethanol and carbon dioxide. And you might notice that that is a key ingredient to generating things like beer and other alcohols. So that's how fermentation works. So we're going to talk a little bit about if we under anaerobic conditions, i.e. there's no oxygen. You might notice that it's really important to have oxygen in these reactions. Well, here we're going to talk about ways we can take pyruvate, which is the end of the glycolysis cycle, and figure out how to get energy when we don't have oxygen. So an important pyruvate step that occurs under anaerobic conditions is the regeneration of NAD plus to pyruvate. So pyruvate will go to lactate and NAD plus by lactate dehydrogenase, and it's basically pyruvate is the oxidizing agent, and it's reduced to lactate. And this process is how we get this is how we use pyruvate to generate energy in anaerobic conditions. Lactate dehydrogenase is a tetrameric isoenzyme, and so it has two subunits, H and M, and they're found in different combinations in different tissues. And so, for example, in heart tissue, H4 predominates, but in skeletal muscle, M is the predominant subunit. So while reduction of lactate allows glycolysis to continue, it increases the concentration of lactate and of H+. So in muscle tissue, and that's when blood lactate reaches a certain level of 0.4 megs per mil, 100 mils, it becomes completely exhausted. That's why you get tired when you work out. And so this is how we get around in anaerobic conditions. This is how we get around the not having any oxygen. We can burn energy. An alternative is pyruvate to ethanol. Again, this is found in yeast. Pyruvate goes to acetaldehyde. Acetaldehyde that's through pyruvate decarboxylase. Acetaldehyde can go to ethanol through alcohol dehydrogenase. And so yeast and a couple other organisms regenerate NAD+ in this relatively simple two-step process. So pyruvate normally goes to acetyl-CoA. So under aerobic conditions, when we have plenty of oxygen, pyruvate takes NAD+ and coenzyme A, sulfated coenzyme A, and oxidative decarboxylation will create acetyl-CoA plus carbon dioxide and NADH. And so that reaction then becomes the entrance to the citric acid cycle, which we'll talk about in another lecture in the future. This is the normal pathway. The other two pathways are alternative pathways. One in yeast, that's their normal, and the other one is under anaerobic conditions. We go to lactic acid. So also stuck next to glycolysis is the pentose phosphate pathway, also called the pentose phosphate shunt. There's numerous other pathways that are names for this pathway. It sits right next to glycolysis, and the important part here is it does provide an entry point. We'll discuss that in a little bit greater detail in the coming slides, but the important part here that I want you to note on this slide, this picture, is that it sits right next to glycolysis. There's exit ways for glycolysis into the pentose phosphate pathway, as well as entry points back into glycolysis, as well as a TCA cycle. Now, why does a pentose pathway exist? Pentose phosphate pathway exist. It does two things. It generates NADPH, which we will talk about later when we talk about anabolism. It's an anabolic pathway. It also generates 5-carbon sugars, which are the precursors for nucleotides. And so it's also important. So it's very, very important it can provide pathways into glycolysis. So it is a catabolic. It can be a catabolic process. Mainly, though, it's a pathway where we can generate anabolic things. We're not going to talk about a nucleotide biosynthesis for very much, so this is why we're going to discuss it here. The important part is it sits next to glycolysis. You can go down this pathway, and with the two main things that we get, NADPH, a reducing agent in anabolic reactions, and we also get the sugars, precursors for nucleotides. All right. Two distinct phases of the pentose phosphate shunt. This is not a great resolution picture, but hopefully you get the idea. There's oxidative phase, non-oxidative phase. In the first initial oxidative phase, we generate NADPH, and in the second non-oxidative stage, we generate the 5-carbon sugar. And the pentose pathway, we're not going to go in, we'll go into some detail on it, but not great detail. It takes place in the cytosol, and from there you can see where we feed into glycolysis. So first couple steps, we're going to be generating NAD+. Step 3, we jump and we then generate the sugars. And then we go, that's the non-oxidative phase. From there we go into glycolysis. That's the important part of this. So the first two steps, oxidative phase, generate NADPH, which is used again as a reducing agent. Then we go into the latter steps, which we generate the sugar, the pentose sugar. So here you can see we're going to take glucose 6-phosphate, obviously the first step in glycolysis. We're going to take NADP+, and we're going to generate a ribulose 5-phosphate plus 2 NADPHs. Then we go through the next steps, and we're going to get down to glyceraldehyde 3-phosphate as we need it. So we can go ribulose, 5-phosphate, glyceraldehyde 3-phosphate. This step, we're not going to go into detail. You don't have to memorize the enzymes and all that good stuff. The important parts that you know about the pentose phosphate pathway or shunt, we've already discussed in detail. So just know that this is how we can get to those steps. And with that, we will end. Thanks for listening. Look forward to hearing you discuss some of the topics as we get going.